BloodWorks North. So Dr. Pavel Davizon-Castillo is also an associate member of BloodWorks Northwest Research Institute. After his medical residency training, he completed his pediatric training at Children's Hospital in Colorado, whereupon he joined their staff as a clinician investigator. And Jose Lopez somehow recruited him to Seattle about a year ago to complement the amazing cadre of investigators there. Pavel has a distinctly unique angle designed to understand why and how platelets are programmed to cause pathologic clotting during disease. So, Pavel, let me just switch that, sorry. You're good to go.
Well, thank you so much. This is really an honor to be here. And if I may, I've been clearly benefited from the legacy of Dr. Davie, because I'm somehow the grandkid of his trainees. I trained with Jose, and the legacy he left at BloodWorks, I mean, I think is great. So the title in your page is a little bit misleading. I'm going to talk about why do we think platelets may be contributing to thrombosis, but also to hemorrhage. So this is something that, the science has led me to investigate this area. So do you know, I'm sure that you guys know, but that platelets, despite their small size, they contain mitochondria, and they really depend on those mitochondria to fulfill their hemostatic function. So we found a few years ago that in response to TNF-alpha, platelets from inflamed mice increase their mitochondrial mass and also their reactivity. This was done in a model of aging where chronic inflammation by TNF-alpha was linked to more reactive platelets. So that was an interesting observation. That was really something that got me thinking about what's the connection between mitochondria, platelet function. So there must be a metabolic component regulating these events. And as we have learned more, and for example, what Dr. Morel today presented, I mean, platelets have become more well known as somehow part of the innate system, and they seem to have some immune functions. So why not to ask the question, is there an immunometabolic response of platelets to inflammation? But this is something we found in mice, so is this happening in humans? So to put this into context, then we decided to study platelets from an age-related hematological disease, myeloproliferative neoplasms. These are a group of hematological diseases in which there's an overproduction of myeloid cells like red blood cells, platelets. There's a subtype that leads to myelofibrosis, and up to 10% of these patients actually can develop leukemia. So this is a neoplasm. But the issue is that these patients, the main cause of death and disability continues to be thrombosis, and to a less extent, hemorrhage, despite being on standard of care treatments. So what's happening here? So in this particular case, I decided to focus on studying just polycythemia vera. 95% of these patients, at least in this cohort, 100% have the JAK2 mutation. This leads to a clonal expansion of these myeloid cells, but also this mutation causes the overproduction of TNF-alpha, similar to the mouse model that I studied in the past, right? And aging and inflammation are two of the main risk factors to develop MPN. So we found that, yes, these patients have high levels of inflammation in plasma, and their platelets contain a lot of more mitochondria than healthy age and sex match controls. So seems to be a correlation, a link between inflammation by TNF-alpha and the platelet phenotype. So we asked the question, is inflammation actually modulating or changing the hemostatic function of platelets? Could this be one of the reasons why these patients, despite being on treatments that actually are usually aimed to reduce the number of cell production, continue to have high number of thrombotic and hemorrhagic events? So we studied a very large cohort of patients in age and sex match controls. And from a 10,000 feet view of their metabolome profile, they're significantly different. But remember, they also have more mitochondria. So does that mean that they actually respire more, they function better? What is this happening? What does this mean? So then we studied their bioenergetic profile doing seahorse assays. But contrary to what we were expecting, these platelets actually have lower oxygen consumption rate, showing actually evidence of significant metabolic dysfunction, mitochondrial dysfunction. And here you can see that their ATP link production or oxygen consumption is significantly reduced. And we corroborated this through our metabolomic studies. They have lower amounts of ATP. So these platelets are in this hypometabolic state. But you might be thinking about, okay, what's the functional consequence? We did all the usual studies, functional studies, platelet aggregation, activation by flow, and all that. What's really, really different here is clot contraction. And this is actually a part of one of the platelet functions that we don't talk a lot, but it's an important function because when a clot is formed, you need to rapidly contract it to make sure that you decrease the obstruction and make sure that you don't cause thrombosis, right? But that contraction also makes sure that the fibrinolytic system comes in and chop the clot in an organized manner, making sure that that clot is a hemostatic clot, not a thrombotic event. So we found that these platelets are really bad. They had defective clot contraction. And this is here. You can see this is one of the many different ways that we tested this. But you can see that the clot is larger. So that means that clot wasn't able to extrude the serum from within and is defective compared to the control. So this is important because we know that a 55% reduction of obstruction in the flow can represent up to 90% obstruction in blood flow. So under the wrong conditions, inflammation, other comorbidities, these can rapidly evolve to become a thrombotic event, right? And there's plenty of evidence in the literature that this is happening in vivo, but also there's plenty of studies showing that this has been documented with inflammatory conditions where there's a lot of thrombosis. But what are the mechanisms? So we went back and interrogated the megakaryocytes from these mouse models, and we found autophagy, one of the processes that could be the culprit. Why? Because this is a very important metabolic hub that sustains metabolism, but also eliminates mitochondria. So I'm sure that you guys are aware, this is a highly conserved pathway that serves as a way to get rid of damaged proteins, mitochondria, lipids, for example. Those cargoes that need to be degraded are targeted into an autophagosome, then they're fused with a lysosome, and these degradation products serve as metabolic substrates. To measure this, imagine that we had this cartoon and you want to see how good your line is producing. So let's say you stop and you just count how many boxes you're making. So the more boxes you make, the higher the flux over time. And then, of course, if there's no change, that line is just not producing. And in this case, LC3B2 is the molecule that we quantify. So yes, these platelets have block autophagy, as you can see here. Controls can increase the production of autophagosomes, but MPN platelets do not have. So very much big picture, summarizing this, we know that these platelets are hypometabolic, they have block autophagy, mitophagy, they're accumulating mitochondria because of that, and they have defective clot contraction. But is this really what's going on? Well, you take platelets from healthy individuals and you block autophagy, you had the same problem, defective clot contraction. But this is inflammation mediated, right? Does inflammation has something to do with this? And because we were thinking that TNF-alpha is an important pathway, we have done this study where we injected mice, we had different mouse models of inflammation, and when we measure an important molecule called syntaxin 17 here in this cartoon is a very important mediator of the fusion of autophagosomes and lysosomes. So what we found is that inflammation downregulates syntaxin 17, and this is really what's causing the blockade in autophagy and mitophagy, causing defective clot contraction. So we're thinking that this is happening at the level of the megakaryocyte. The megakaryocyte is downregulated syntaxin 17, is packing platelets with less syntaxin 17. And these platelets are handicapped from the get-go. They're really, really set for failure when it comes to autophagy and mitophagy, and that's causing this hypometabolic defect in platelets. So we're thinking that this is one way, this is an example of how inflammation causes this. This is an example of immunometabolic regulation of platelet function. And what we think is going on is if you have this defective clot contraction, then you have a higher obstructiveness that can lead to thrombosis, or you can have a clot that is weaker and can easily dislodge and cause the thromboembolic event, or it's a clot that is easily dislodged and disrupts primary hemostasis, causing hemorrhage. So I just summarized to you 6 years of work in a few minutes. I'm sorry, believe me, there's a lot more than this. I want to thank Guadalupe, Dr. Guadalupe Rojas. She's driving this project, and please stop by her poster, and I'm happy to take any questions. Oh, yeah.
What about the role of myosin in platelet clot retraction?
Myosin, well, that's definitely another factor that can affect contraction. What we know, for example, in NYH9 defects is that there's a problem with the protein, right? It's not there, the right protein. So what we're thinking here is that it is the energy required to provide for the contraction to happen that is disrupted in this particular pathway. So what we think is this is probably a generalizable mechanism of disease because of thrombosis and hemorrhage in inflammatory conditions. But yeah, that's a completely different possibility.
I have a question out of curiosity. So comparing platelet derived from bone marrow megakaryocytes or lung megakaryocytes, would you suspect they have different mechanisms in this?
I don't know. I was going to ask Craig about, should we do something about it? Yeah, I wouldn't be surprised. Yeah, thank you.